This is a presentation on phosphoproteomics for BS 3336 Proteomics Workshop. Proteomics is the scientific discipline which studies proteins and searches for proteins that are associated with the disease by means of their altered levels of expression or post-translational modification between control and disease states. Following the release of the human genome sequence data in 2004, humans are considered to have 19,599 genes that encode for proteins. Alternative RNA splicing and post-translational modification may result in one or more proteins or protein fragments. As a consequence, the proteome is far more complex than the genome. The term proteome was first used by Wilkins to describe the protein complement to the genome. Since its first use, its meaning and scope of the term has narrowed through the years. The host of post-translational modifications, alternative splice products, and proteins intractable to conventional separation techniques has each presented a challenge towards the achievement of the classic definition of the word. Phosphoproteomics can identify, catalog, and characterize proteins containing a phosphate group as a post-translational modification. It can identify the site of phosphorylation in proteins and gather knowledge on how it brings about metabolic and biomolecular changes in a cell. It also studies various kinds of kinases and phosphatases to identify the site of phosphorylation and dephosphorylation in proteins. Protein kinases contain a highly conserved catalytic subunit which transfers the gamma phosphate from nucleoside triphosphates, often ATP, to one or more amino acid residues with a free hydroxyl group in a protein substrate side chain. This results in a conformational change and affects protein function. Phosphorylation is a key reversible modification. The reverse reaction called dephosphorylation is catalyzed by protein phosphatases and it produces a free phosphate group. The target amino acid is usually serine, tyrosine, or threonine residues in eukaryotes and histidine and aspartic acid residues for prokaryotes. Protein phosphorylation on serine, tyrosine, and threonine residues is the most prevalent post-translational modification and occurs in more than one-third of cellular proteins. In the human genome, there are 518 protein kinases and about 150 protein phosphatases that regulate phosphorylation networks and control biological processes such as proliferation, differentiation, and apoptosis. The enzymes for phosphorylation and dephosphorylation fall into two broad classes characterized with respect to substrate specificity, mainly serine and threonine specific or tyrosine specific. There are also protein kinases that phosphorylate other amino acids, such as histidine kinases that phosphorylate histidine residues. Protein kinase and phosphatases work separately and in a balance to regulate the function of proteins. The phosphorylation process regulates complex formation, protein function, subcellular localization, protein degradation, cell signaling networks, cellular processes such as metabolism, transcription, cell cycle progression, cytoskeletal rearrangement, cell movement, apoptosis, and differentiation. They also function in embryonic development, physiological responses, and in the nervous and immune system. With all of these modification results, it is assumed that up to 30% of all proteins may be phosphorylated, some even more than one time. Compared to expression analysis, phosphoproteomics provides two additional layers of information. First, it provides clue on what protein or pathway might be activated because a change in phosphorylation status almost always reflects a change in protein activity. Second, it indicates what proteins might be potential drug targets. While phosphoproteomics will greatly expand knowledge about the numbers and types of phosphoproteins, its greatest promise is the rapid analysis of entire phosphorylation-based signaling networks. Phosphoproteomics is more complicated than simply measuring protein expression. First, the presence of phosphorylation has to be detected, following which the site of phosphorylation has to be determined. There are 6 methods available for the study of phosphoproteomics: radioactive labeling, phosphoprotein staining, immunoprecipitation, mass spectrometry, 2D phosphopeptide mapping, and protein microarray. Radioactive labeling of proteins with 32P or 33P is the oldest, but still it is one of the most sensitive approaches for detection of phosphorylation. Under appropriate conditions, the phosphoryl groups of 32P or 33P are enzymically added to the proteins. The phosphorylated proteins are then detected by autoradiograph. Because of this, radioactive labeling is able to detect all types of phosphorylation. However, only small amounts of radioactivity will be incorporated in proteins. Moreover, the quantity of the isotope and duration of labeling is important as it can artificially induce changes in phosphorylation. Safety issue is of a great concern. In 1973, phosphoproteins was detected through a phosphor-specific dye called cationic carbocyanine dye, which is stained in acrylamide gels. It has been widely used for over 40 years. However, it has low sensitivity with several orders less than 32P radioactive labeling. Another form of staining is using methyl green or rhodamine B. It helps to detect the released inorganic phosphate by forming a precipitation with calcium producing an insoluble complex. Detection sensitivity of the staining method is still very poor, as a protein containing roughly 100 phosphoserine residues is required for detection. Besides low sensitivity, phosphotyrosine cannot be detected as it cannot be hydrolyzed. Currently, ProQ diamond has increasingly become the first choice of phosphoprotein dye. It is a fluorescent dye and it is suitable for the detection of phosphoamino acids directly in acrylamide gels. The sensitivity of ProQ diamond staining has been improved significantly and is down to 1 to 16 nanograms. It can be used in combination with a total protein stain. It also has greater convenience and safety of handling. The stain is also specific to the type of phosphoamino acid present. Detection is easy with visible light scanning or a visible transilluminator. The emission and excitation maxima for this stain is approximately 555 to 580 nanometers. However, in complex protein samples with thousands of protein species resolved by 2DE, some non-phosphorylated but rather abundant proteins may be also weakly stained. In 2001, the first documented phosphoantibody was an anti-tyrosine antibody which broadly recognized proteins containing phosphotyrosine. After that, there has been a rapid development in production of phosphoantibodies. A large amount of phospho-specific antibodies targeted to different amino acids at distinct sites in proteins have been produced, and are widely used in basic and clinical research. Antibodies are phosphoamino acid specific and hence is necessary to perform several parallel immunoprecipitation reactions to find out the specific amino acid. Antibodies can be used not only with extracted proteins and peptides but also intact cells and tissues. It has high specificity and sensitivity. It can be used to enrich and purify phosphorylated proteins and peptides. However, the major decisive factor for selection of antibodies is their specificity in detection of a phosphoprotein. Therefore, the quality of antibodies become the key concern on their applications. Mass spectrometry is one of the most modern techniques for detection of phosphorylation. Detection of phosphorylation by MS has been based on mass spectrum generated by trypsin digested peptides. In spectrometer, there is an ion source that causes molecules to become ionized. These are then transmitted to a mass analyzer where different ions separate according to their mass. It is then detected by a detector and generates signal intensity proportional to the number of ions. The data system then shows the intensity of each mass over charge. Firstly, we need to stain the cells for MS. SELECT is an in vivo approach where two cell cultures are labeled differently with heavy arginine 13C heavy isotopes or normal arginine 12C light isotopes. When cells are grown in the medium, they incorporate heavy arginine into all of their proteins. Another type of isotope labeling is iTrack. iTrack uses stable isotope labeled molecules that can be covalently bounded to the N-terminus and side chain amines of proteins to determine the amount of proteins from different sources in a single experiment. These samples are then pooled and usually fractionated by nano-liquid chromatography and analyzed by tandem MS. The proteins now have to be extracted from the cell culture to be denatured for analysis. Cells are lysed and protein is purified through acetone purification. Precipitation has an advantage over dialysis or desalting methods as it enables concentration of the protein sample as well as purification from undesirable substances. One disadvantage of protein precipitation is that the proteins may be denatured. Next, it is the generation of peptides. Protein digestion is when three important processes occur, and mainly reduction, alkylation, and trypsin digestion. For reduction, TTT is added and incubated at 30 degrees Celsius for an hour to reduce disulfide bonds in cysteine residues. For alkylation, IAA is added and kept in a dark room for 30 minutes in room temperature to prevent the formation of disulfide bonds. Trypsin is then added at a ratio of 1:100 overnight at 37 degrees Celsius to break proteins into peptide fragments. Phosphopeptide enrichment allows maximum amount of phosphopeptides to be collected. Studies have shown that phosphopeptide enrichment has been amplified by coupling more than one method. For every enrichment step used, desalting is required to prevent the salt from interfering with the signal in LC-MS analysis. Immunoprecipitation is a method used in the enrichment of phosphopeptides. It involves and employs antibodies raised against phosphorylated amino acids. Antibodies are phosphoamino acid specific, and hence it is necessary to perform several parallel immunoprecipitation reactions to identify the specific amino acid present. Immunoprecipitation is very useful to observe amino-specific phosphorylation one at a time. A second method, IMAC, employs a matrix composed of resins with associated metal ions. These metal ions are positively charged and hence can catch negatively charged phosphate groups along the column, which are then eluted. SEX separates peptides by their solution charge state. When performed at a pH below the pKa for the acidic amino acids, only phosphorylated amino acids can contribute a negative charge, and a given phosphopeptide will elute earlier than its unphosphorylated form, concentrating phosphopeptides in the earliest fractions. The next method is based on the selective interaction of phosphopeptides with porous titanium dioxide microspheres via binding at the TiO2 surface. Phosphopeptides are trapped in a TiO2 pre-column under acidic conditions and are eluted under alkaline conditions. TiO2 resin typically binds phosphopeptides with a high affinity and the preferential enrichment of singly phosphorylated peptides with TiO2. For LC-MS, peptides are loaded into autosampler and machinery. Here is a video to explain the process.
Agilent's LC-MS ion sources use our patented nebulization technology and high-volume counterflow drying gas. Together, they reduce noise related to incomplete drying of solvent droplets and keep the sampling capillary and ion optics cleaner for reduced maintenance. A thin skimmer aperture, carefully matched hole size, and short capillary to skimmer distance reduce beam broadening. An octopole ion guide provides better ion transmission over a wider mass range. Patented lenses enhance high mass ion transmission and increase sensitivity over a wider mass range. The first quadrupole mass filter allows only ions of the target mass to pass through. The hyperbolic shape of the rods enhances ion transmission and spectral resolution. In the hexapole collision cell, precursor ions strike collision gas molecules, generating product ions and neutral fragments. Linear axial acceleration and high collision gas pressure simplify operation and ensure fast, sensitive MS-MS without cross-experiment memory effects. The second quadrupole serves as a mass filter for the product ions produced in the collision cell. For quantitative analysis of a target compound, the second mass filter is operated in a selected ion monitoring mode. In the detector, the conversion dynode operates at 10,000 volts to improve sensitivity. Because the conversion dynode is off the main axis of the ion path, neutral molecules miss the dynode, thus eliminating neutral noise. A secondary dynode helps extend the useful life of the electron multiplier. The electron multiplier has a long life, but it is also easily replaced. The Agilent 6410 triple quadrupole LC-MS establishes a new standard.
MS provides a high speed and high sensitivity means of detection for phosphorylation. Introduction of MS has significantly advanced the research in protein phosphorylation. It may be applied not only for detection of phosphorylation, but also the identification of phosphorylation sites and its quantification. However, there are several inherent difficulties for the analysis of phosphoproteins. Firstly, signals from phosphopeptides are generally weaker as compared to non-phosphorylated peptides, as they are negatively charged and poorly ionized by Ms performed in the positive mode. Secondly, it can be difficult to observe the signals from low abundance phosphoproteins of interest in the high background of abundant non-phosphorylated proteins. To overcome these drawbacks, enrichment of phosphoproteins or phosphopeptides before MS is necessary. The results obtained from LC-MS is then analyzed using data search-based or computer technology. For analysis of SILAC, the differentially labeled peptides having the same amino acid sequence are detected in MS spectrum, and the relative abundance of their peptides can be compared by calculating their ratio. For the analysis of iTrack, the reagent consists of reporter regions of different molecular weights. Each tag generates a unique reporter ion in the MS-MS spectra, and the relative abundance of their peptides can be compared by calculating their ratio. No single method is sufficient for comprehensive phosphoproteome analysis, and the combination of different approaches for enrichment can improve the comprehensiveness of phosphoproteins. The combination of various strategies such as LC-MS, 2-DE gels, and peptide microarrays can increase the reliability and comprehensiveness of the data we can gather from the experiment. 2D phosphopeptide mapping is a traditional biochemical method for identification of protein phosphorylation sites. Phosphopeptide mapping of these phosphorylated residues allows investigation into the positive and negative regulatory roles these sites may play in vivo. In addition, phosphopeptide mapping can uncover the specific phosphorylated residues and hence kinase recognition sites, which help in the identification of the relevant kinases or phosphatases. This method allows easy comparison, as it's the most common way for confirmation between wild type and mutant proteins. However, it is time consuming and care must be taken when labeling the cells with radioactivity. It can only study single proteins and cannot be applied to large-scale identification of phosphorylation sites. Arrays based on application of protein microarray offer the potential of profiling the proteome without employing separation techniques and evaluating protein biochemistry in a high throughput and systematic manner. The substrates were immobilized onto nanowell protein chips, and phosphorylation events were identified by the addition of 33P gamma ATP. The reversible phosphorylation of proteins regulates almost all aspects of cell life, and hence any abnormal activity in this phosphorylation process may affect us in a dire way. Mutations in particular protein kinases and phosphatases give rise to a number of disorders, and many naturally occurring toxins and pathogens exert their effects by altering the phosphorylation states of intracellular proteins. One such example of a protein that can cause a disease when phosphorylated is the heart muscle protein called myosin binding protein C. I hope that this presentation has better helped in your understanding of phosphoproteomics. Thank you for your time and attention.